Okay, you guys, I am back. So we had just finished up talking about bacteriophages in the lytic and lysogenic cycle, and I had talked about it with this picture and started to go on to the animal viruses before I had issues, but I forgot I had to go back and fill in the blanks about the lysogenic cycle. So that's what we're going to do here. So the lysogenic cycle down here, all right, replicates without destroying the host, which is what we just saw, and remember, allows the host to continue to replicate the viral DNA as the host cell replicates. The viral DNA is incorporated by genetic recombination, so that means two sources of DNA recombine together, and so you have part bacterial or host DNA and part viral DNA. And then it makes what's called a prophage, so that integrated viral DNA is a prophage, and every time the host cell divides, it copies the phage DNA and therefore passes it to the daughter cells. And this type of phage that does both of these, the lytic and the lysogenic cycle, are called temperate phages, whereas the bacteriophages that just do the lytic cycle are the virulent phages. Okay. All right, so now we are ready to go on with the animal viruses. So animal viruses, basically, most of them have viral envelopes, membranous envelopes around the capsid, and they're classified as DNA or RNA, and whether or not that is single-stranded or double-stranded, just like we talked about before. So, as we said, many viruses infect animals that, and those viruses have membranous envelopes, and they have on the surface of those envelopes what we call glycoproteins. That means it has a carbohydrate with the protein on the envelope, and those the glycoproteins help to bind the virus to bind to specific receptors on the surface of the host cell. So let's look at a typical RNA virus. And so this is figure 18.8, as it says on the top, and the title is Reproductive Cycle of Enveloped RNA Virus. So they have an envelope, so that's what we see here around here. So this is the envelope. These little guys right here coming off of them are the glycoproteins. Then right underneath there, you have the little balls here are the capsid. So this right here is the capsid. And then remember, capsids always enclose the genetic material, and this is an RNA virus, so here's your RNA. So that's what we have as the structure of the virus that's going to infect this host cell. So now if you notice from this virus going to the host cell, the first step here is that, look at what the only thing that enters into the host cell is the inside, the capsid with the RNA. And so the envelope around there, so here's the capsid around here and the RNA, the envelope doesn't go in. So what is the envelope for? So these glycoproteins on the surface here, the envelope with these glycoproteins help. What they do is they bind or bonds to receptors. Binds to receptors on the cell membrane. The host, I should say, host cell membrane. So what happens is there are proteins embedded in the surface of the host cell, and the virus comes over here, the glycoproteins can bind to that, and then they fuse and open up like this. So this right here, so this is the envelope, fuses with the host cell, and then everything that's inside, which is the capsid, goes into the host cell. So this becomes part of the host cell membrane here by that way. So then it breaks up the capsid. And so what you end up having is here's your viral genome exposed. All right, so that is RNA. So then what this host cell does is take RNA, and remember RNA, if you have the order of bases in RNA, A, U, G, C, A, U, so on and so forth, of RNA, we can build from that, the host cell can take the RNA and build a template RNA from that. All right, so this is the template right here. And it makes copies of the RNA, but they're going to be opposite bases here. So when we say that we have copies here, so this is the copy, all right, from the RNA, what that's going to do is, so that copy, so this is going to be like the original, the template is like this, and then the copy is going to be like U, A, C, G, U, A. So that's the copy. So it's not an identical copy, but it's the complementary basis to that identical, that original template. So then notice here, what happens is we go over here, and then we copy it some more. And so this little guy here comes over here, all right, and we make copies of it. And the copy can be used to actually be the new strand or the new genomic or genome for the new virus. How that is done is going back to this example up here. So you had your original template, which was in green, and then here's your, they call it the complementary mRNA. And so when the copy, the complementary DNA, which is what that copy is called, the complementary parts, or sorry, not DNA, RNA comes over here. So this little guy here, that's this guy here, it's UAC, GUA, and whatever else it is. And a copy of that is made. So then notice here when we go, so this goes, it's going to go back to what the original code was. So we have AUG, CAU, which is what was originally. And these copies are going to then go and be the new genome for the new virus. All right, so that's one thing that the mRNA is used for. So that's the first pathway. The second pathway is over in this direction, and notice the complementary mRNA that was made. So this is the blue. All right, here it was used to make the original sequence so it can go into the new viruses. The complementary mRNA is actually used in protein synthesis. So that's what we see here. So this is made, this can actually go and be used by bound ribosomes on the ER. It's made by, if it's used and makes proteins on bound ribosomes, it helps, what it does is code for making more glycoproteins. So notice here you have the ER, the endoplasmic reticulum, and the endoplasmic reticulum has those ribosomes on it, and an mRNA is used by those ribosomes, and it makes more of these glycoproteins. So then, the second place that the mRNA can go is to the free ribosomes and be used to make the capsids for the new virus. Because remember, the virus overall is going to use the host cell to make new viruses. So basically, a virus is the capsid and the protein and the genetic material, which in this case is RNA. So we have these two things done, and then remember the endomembrane system way back from the cells that vesicles bud off of the ER. So here's the vesicle, and it has the glycoproteins on the inside there. And then remember what those vesicles did? Some of them can fuse with the cell membrane and excrete its contents. Well, when it does that, the vesicle membrane becomes part of the host cell membrane. So guess what? On the outside of the host cell now, you have all these glycoproteins, which really stem from the virus. And so on the outside of the host cell, from the virus binding with it to in the first place, and and the vesicles binding with it, the outside of the host cell has all of these glycoproteins that are very similar, or are the same, I should say, as the virus here. And so then what happens is these capsid proteins, they assemble around the RNA, the genetic material, and they leave the cell just like exocytosis, budding off the membrane, buds off. But remember, the membrane, though, now has these glycoproteins, and so now when it buds off, you have your membrane around with the glycoproteins. You have your capsid now, and you have your genetic material, which in this case is RNA. And so you have a brand new virus that looks exactly like that first virus right there, and it has used this cell to replicate. And so it can do this. They're showing you one virus coming off, but then they can make lots of viruses, and lots of viruses bud off, and you go from one to many, and that's how they replicate. So for animal cells, the envelope with the glycoproteins are helpful in entering the host cell. The glycoproteins bind with the specific receptors, and that's one of the reasons why a virus can't infect every single cell, is because these glycoproteins are very specific in the receptors that they bind to, and so therefore there's very specific host cells that they infect, which is why another reason why viruses can't infect every single type of cell on Earth. Okay, so that is a typical animal virus. All right, so now we're going to look at another example of an RNA virus. So it says here RNA as genetic material. The broadest variety of RNA genomes is found in viruses that infect animals. So there's a lot of RNA viruses that infect animals. And a specific type of RNA virus, we talked about one up here. So this is one way in which an RNA virus can reproduce. A second one is through what we call retroviruses. And retroviruses use an enzyme. How do we know it's an enzyme? It ends in ACE. They use an enzyme called reverse transcriptase to copy their RNA genome into DNA. So it's the opposite of transcription. So just a quick little review. Remember that you have your double-stranded DNA. DNA, remember, is used to make mRNA in the process of transcription. All right, so reverse transcription is going not from DNA to mRNA, but the opposite, going from RNA to DNA. So they copy so it's RNA to DNA. All right, and HIV is the most popular or common retrovirus. It's not the only one. HIV is human immunodeficiency virus, and it is the virus that, when you're infected with it, that can cause the disease that we call AIDS. All right, and so when we look at the virus here, the virus of a retrovirus here has again, the viral envelope. It has the glycoproteins, which remember binds to the receptors on the host cell. Inside here we have our capsid, which I outlined in purple. And then here we have 2 RNA strands. All right, so HIV actually has 2 RNA strands as their genetic material. And then in red, I put here these little balls here are the enzyme reverse transcriptase. So this virus comes with its own enzyme, all right, and then can make the opposite of transcription. So if we look at what happens here, let's focus on the picture first, and then, like I said, we'll go back to, like we did before, I should say, go back to the bullets up above. So this picture is figure 18.10 in the book, and it's the reproductive cycle of HIV, a retrovirus. So I'm going to show you how it's a little bit different than just the normal RNA virus that we looked at in the previous picture that just used the host cell to make more RNA for the viral genome. So this virus enters into the host cell just like the last one we looked at. So the glycoproteins bind to receptors on the cell membrane. The capsid goes in. The capsid is broken down. And so now we have the reverse transcriptase, which is here in red. And then you have your host cell's DNA in green. Okay, I'm sorry, RNA, not DNA. And so then here we take one of those pieces of viral RNA, the virus's genome. So in green here, that's the viral RNA. Here's the enzyme reverse transcriptase. So what does reverse transcriptase do again? Remember, it takes it and goes from RNA to DNA. So that's what we see in the next picture. So here is your RNA. That's the same thing. The reverse transcriptase is now going to use that RNA as a template to make a strand of DNA, but it's a single strand, and you know our DNA is double-stranded. So this is DNA right here, and then the green up here was the viral RNA. And it can't do that unless it has that enzyme, which it comes with it. Okay? So then what happens is then once we have one strand of DNA, just like in DNA replication, we can make another strand by complementary base pairing. So now here we have, this is our double-stranded DNA. All right, so now look at the picture. What's going on? So that double-stranded DNA enters into the nucleus where our DNA is. So that's where it says chromosomal DNA right here. So this is our DNA right here, chromosomal DNA. And what happens is this DNA that now codes for viral proteins, because we made a DNA strand from the virus's RNA. So we have the complementary bases, and basically it codes for viral proteins. Gets inserted into our genome. So that's what we call a provirus. A provirus, this is, let's write this here, DNA that codes for making viral protein inserts into host cell DNA. All right, and I want to make a note that this is permanent. So then what happens is this DNA that's not part of our DNA, it codes for making these viral proteins, stays in us. And so what happens is that we can make and transcribe it at any time. So then what happens here, from here to here, when we can go through protein synthesis and make mRNA. So this is transcription. So we turn this gene on, we make an mRNA strand, so RNA polymerase binds and makes the mRNA strand here. So this is mRNA. So then look at what happens is it goes in two different directions. So the mRNA, this can actually be used as so this is one direction. It's used for the next viral generation, so it's the RNA genome. It'll be actually incorporated into new viruses that the cell is going to be making. And the second pathway is the mRNA can be used to go through translation. So it can be used, this step here is making proteins. So this is translation. Except instead of human proteins, they're making viral proteins, and they're made in just the same way as that last picture. So some of the mRNA goes to bound ribosomes on, doesn't say here, but this should be the ER right here. So there's bound ribosomes on the ER, and the mRNA comes over here and is used to make proteins, and here it's making the glycoproteins, and so they're on the surface, interior surface of the ER, which when the vesicles bud off and then fuse with the membrane, those glycoproteins will now be on the surface of the host cell. And then the mRNA can make the capsid proteins. And so what we end up with is these capsids surround the RNA, and the capsid proteins surround the RNA and make a new capsid, and that capsid leaves the cell through exocytosis, but since the cell membrane has these glycoproteins on the surface, it ends up being just like this host cell here. And so what happens is this host cell doesn't die, it stays alive, always has that provirus in it that has the code to make viral proteins, and so this process of making new viruses can continue to happen while this cell stays alive, and the cell can continue to make new viruses here. And so this picture is actually the picture of this happening here where it's budding off. So you can see here, and that's the new HIV. And then the same thing up here shows the actual picture of this process of it actually entering into the cell. And so HIV is a little bit different than other RNA viruses, like the previous pictures the previous picture we looked at. So here's the other RNA virus that just uses the host cell to copy its RNA and therefore make new viruses, but the genetic material is not incorporated into the host cell's DNA, whereas the retrovirus and HIV specifically as an example stays within the host cell's DNA and can continually make new viruses that way. All right. So because of this, viruses are not necessarily considered living organisms. They don't fit our definition of living organisms because they cannot survive on their own. They need a host cell. They depend upon a host cell to reproduce. They can only reproduce within those specific host cells. And so, but viruses are very savvy. They can mutate very quickly, so their genetic material can change, and they have survived for many, many, many years. All right, so, but they do require a host cell to replicate. And that, my friends, is 18.1. So like I said at the beginning of the video, if at the, on part 1, that I accidentally gave you the wrong guided reading, but I will put it on my website if you want to get a head start. But in terms of homework due on Wednesday, I will cancel that, and you will eventually have to do it. So if you want to get a head start on it, you can check that out. All right, and then if you have any questions on any of these, jot them down on in the margins and the notes and so on, because that's what we'll start out with on Wednesday. I'll see you Wednesday. Have a great 4-day weekend. Just kidding, I forgot to go back up here and do the fill in the blank. Sorry, guys. So the viral DNA that is integrated in the host genome is called a provirus. So that's what we wrote here. And a provirus remains permanent, so we talked about here. And how it makes new viruses is the host cell's RNA polymerase transcribes the proviral DNA into RNA molecules. And so these RNA molecules have 2 pathways, so if they function both as mRNA for synthesis of new viral proteins and as genomes for the new virus particles released from the cell. So here's the genome, and here's the using it for protein synthesis. All right, so that is the end. Have a nice weekend.